Very happy to introduce our next speaker, Dr. Verdin, the leader of Buck Institute for Healthy Aging, and so much more than that. Thank you so much for being here. Extremely resilient to stress. Thank you. Wanna put it on? Yeah.
Thank you, Evelyn. Good morning, everyone. Delighted to be here. Let me start by thanking Evelyn for her leadership of this meeting, and Morten and Alex for actually including a longevity medicine day at this meeting. I think as a physician scientist, it's actually amazing to see that there's a true interest in really bringing all of these discoveries and all of this work to the clinic. And as Neers has said, it's not going to be easy, but that's not a good reason not to try. And I think we're really excited to be part of this. I'm going to be talking about a sort of just a scientific finding today. So this is not a general talk, but more a specific area in which the lab is currently moving. As we are moving as a field, we have actually been remarkably successful at discovering novel molecules that are actually targeting the aging process. And I've listed here what we call the longevity drug pipeline. And we just heard about metformin and rapamycin, but there are many other drugs that have been shown to... Oh, did I knock something off here? Okay, we're back. A number of drugs that have been identified as having an effect on lifespan in a variety of animal models, including one that's not talked about enough, I believe, is acarbose, which also has an effect on glucose absorption, senescent cell clearance, senolytics, and so on. And today there are over 100 interventions that actually can extend health span and lifespan in mice. So we're not short of potential targets and drugs in the future, and this is quite exciting. However, as it's been alluded to by Neer, there are a whole series of issues in terms of conducting trials in humans, and one of the biggest one is nobody's planning to do a lifespan study in humans with any kind of experimental drug. So hence the need for what we call biomarkers of aging, surrogate markers that would allow you to really measure the effect of your drug and to predict that this drug is going to have an effect on lifespan. So I've listed on this slide a number of so-called ideal biomarker of aging that we will need in the future to conduct this work. So it should be hopefully a reflection of a ubiquitous aging-related process. Ideally, it would be universal. It should work across tissues. As we are limited in our ability to sample, most of the times, as you know, we sample the blood. So that the blood should be reflective of what's happening in different organs. It should be based on the cellular, subcellular level process. It should be predictive of health span or lifespan, or both. And more importantly, and this is something that really has not been shown very much, it should vary in response to your intervention. And finally, it has to work in people, in men, in women, and in people of different ethnic backgrounds and racial origins. So obviously, we're not there, and maybe the data I'm going to show you, we're actually quite far from there. And so there's a lot of interest in the field in trying to really further this work. Now, one of the big problems that we are confronted with, and I'm sorry, today I will be identifying a problem more than really providing a solution, but is the fact that the major organ that we're sampling is the blood. And we know it is made out of at least 50 different cell types. And therefore, when we sample in the blood, and we know many of these blood subtypes actually vary during the aging process, and I'll show you some evidence for this. So typically, we separate the blood. Almost all of the cells that are present in the blood originate from a single hematopoietic stem cell, and which can differentiate into a lymphoid precursor or a myeloid precursor. Lymphoid would be the adaptive immune system, B cells, T cells, and the myeloid would be your macrophage, monocytes, mast cells, basophils, and so on. These two different forms of the immune systems are very different, the innate being the rapid response, the adaptive being the response that can be educated by vaccination and leading to what we call an anamnestic response. And we've done a lot of work over the years primarily on CD8 T cells, and so I'll be sharing with you some work that we've done on this. So the CD8 T cells would be, is this a pointer? Okay, the CD8 T cells would be right here at the bottom. It's part of the T cells, the adaptive immune response. Now, there's something really unique about CD8 T cells and CD4 T cells is that they both include an obligatory developmental step that happens in the thymus, which is shown here on the left. And the thymus, interestingly, is one of, like the ovary, is one of the earliest organs to show an age-related phenotype. By the time you are 50, most of us have actually lost our thymus. It's completely involuted, it's been replaced by fat, which means that this differentiation step into naive T cells really happens until you are, actually it happens mostly during early life, but it's essentially done by the time you're 50, which means you're relying on these naive T cells which migrate in the periphery to differentiate into a series of steps. And I've shown here on the upper left the naive CD8 T cells. It becomes eventually an effector T cells and eventually a true memory T cells. So there's a limit there in terms of our aging, because by the time you're 50, you're left with a pool of naive T cells which are not going to be regenerated. So they are maintained by a process called homeostatic proliferation. And so I just want to share with you some data that illustrates what happens to these cells as we age. And to do this, we've relied on a novel form of flow cytometry. So flow cytometry allows you to categorize immune cells in the blood. Your typical flow cytometer has 6 to 7 colors. We now have available in our lab and others a new form of spectral flow cytometry, which actually encompasses up to 45 colors. And so just imagine the increase in dimensionality that you can do with this type of approach. You can actually really, in 2 or 3 panels, we can map the relative abundance of more than 50 different cell types within the blood. And so we've done an experiment I'm going to show you in 1 minute that encompasses humans between 20 and 82 years old. They were equally divided between males and females, and including also equally represented in terms of CMV, cytomegalovirus serostatus. This is a very frequent infection in humans, which might have some impact on the immune system. And so when you do this, on the left side, you can see what we call a dimensionality reduction plot, which allows you to take all of these variables, all of these 40, actually it's more than 44, it's 3 panels of 44, so there's 120 different colors, and reduce this into a 2-dimensional plot. And you can see we can now really identify many different cell types in the blood, including your monocytes on the left or your B cells and so on. And the first thing that we see there is, as a function of aging, we see a relative increase in monocytes during aging. That's been known for many years. And also a relative decrease in the CD8 T cells and the gamma delta T cells, reflecting this lymphoid to myeloid shift that I've talked about earlier. However, we see even bigger difference when we now focus just on the CD8 T cells. So there's another one of these UMAP plot on the left, and you can see what we used to call the CD8 T cells that can actually be divided, and we have at least 14 different populations. And by the way, using this type of technology, we're able to identify some novel populations that actually increase during aging, and we are currently in the process of characterizing. So there's a lot of biology there that I won't have the time to dig into, but I hope that the plot shows you the level of resolution that we can attain, which was not available as recently as 5 years ago. When you do this and you start looking at selective population, you can see that the naive T cells actually show a pretty precipitous decrease during aging on the right side. And when we look at central memory T cells, we see exactly the opposite. they proportionally increase. Same for the effector memory. And so this goes back to what I told you about the differentiation. You start from a limited pool of naive T cells, which as we age, encounter their antigen and transition to becoming effector memory or more differentiated. And so the naive T cells pool decreases and the effector T cells increases. Now the question that we ask is, do these change in populations have a bearing on some of the clocks that we are using today? Because obviously the blood is not a static process, and hopefully you're convinced now that there are big changes happening during aging. And so I want to talk a little bit about the epigenetic clocks, which have emerged as one of the major markers that the field is focusing on for their potential to measure biological aging. And if you're not familiar with this, just one word of introduction. I'm sure we're going to hear a lot more about them throughout the meeting, but they are essentially tools that allow you to start from either chronological age or clinical characteristic of patients, and to regress these variables onto DNA methylation measured throughout the genome. And so this is done using a variety of regression tools and machine learning. Now, the beauty of this is that once you have the data, you can then go back in the opposite direction. It starts from DNA methylation itself and actually predict what is the chronological age or the clinical characteristics of the patient. So this is the basis of this. And obviously, this whole work has been pioneered by Steve Horvath and his colleague, Morgan Levine, and others. And we now have a whole variety of clocks. Actually, it's about one a week at this point, popping up of different ways in which we can measure biological aging. And so we asked a very simple question. In many cases, these clocks actually were based on data obtaining from the blood. And the simple question we ask is there an effect of variation in the amount of different population in the blood on these clocks? And so to do this, I just want to show you a preliminary data in which we use 7 donors. And in each, we can focus on the whole blood on every population. So we focus on CD8 T cells and ask a very simple question. Now, I always assume that every cell within the blood had the same epigenetic age. And so to measure this directly, we sorted the naive T cells, central memory T cells, effector memory T cells, and the final differentiation product, and then measured DNA methylation in each of these subsets. And what we saw was actually pretty astounding to me, because if you look at the naive on the left all the way to the EMRA, so from left to right, this is the further differentiation of these T cells. You can see that in any given individual, on average, the delta is 25 years. So your naive T cells are actually, on average, 15 years younger than your chronological age, and your finally differentiated T cells are actually a little bit older, about 5 years. So really clearly, I showed you also in a minute ago that naive T cells decrease precipitously. So could it be when we're measuring aging in the blood using the clocks that we're simply measuring a change in the relative proportion of naive T cells versus other cells? And I should say, we have since done a clock measurements in pretty much every cell types in the blood, and the delta actually get even bigger. So it's not limited to CD8 T cells. We see it in other blood types as well. Now, what's interesting is, so you might think, well, the clocks are just a measurement of population change, but there's another data here that's also interesting, is that even though there are differences between different subtypes, there is still a picture related to aging. And what I'm showing you here is that if you look at the donor age, I'm showing you the same data presented in a different way. If you look at the donor age on the x-axis and the predicted age on the y-axis, you can see that the naive T cells, even though they look younger, they're below the intermediate axis, they're all younger, but there is still an aging-related signature. That is, if you take a naive T cells from a young individual, it still looks younger than an older individual. And again, the same thing in every one of the populations. So this data indicates that the current clocks apparently are a mixture of two variables, something that we call intrinsic aging, which is something that happens in naive T cells as they age. It also happens in central memory T cells as they age. But in addition, there's probably another variable, which is the change in relative proportion. So the interim conclusion here is that the current clock are likely to represent a mixture of CPG that vary across distinct differentiation states and of CPG that change as a consequence of aging. Now, this suggests also that change in differentiation state can give the illusion of aging acceleration or reversal. And this is important because there's been a lot of talk about age reversal and the first age reversal in humans. I hope to convince you that I'm extremely skeptical about these data in terms of what they mean, because you will see the example in a minute. So our standpoint is that there's clearly a need to separate these two variables and to generate a clock that measures ages intrinsically. So we started this with a talented student in the lab, Alan Tomusiak. We went back, and there's an enormous amount of DNA methylation data available on a variety of sites. He downloaded what we think is one of the largest collection of CPG sites from 14,000 samples. And Alan had the idea to do something very simple, which is what happens if we would remove all of the CPGs, the methylation sites, that we find change during differentiation of CD8 T cells. And so this is what he did. So we went from 350,000 CPG to about 84,000 CPG. And we used these sites then. The idea was to eliminate the signature that's linked to differentiation and try to determine, is there still an age-related signature? So we used this as a training set, and this generated a new clock. And so this new clock, you can use it on two different things. You can first use it on your data set that you use to generate the clock. This is sort of a highly biased way to do this, but the good news is that the clock works, as you can tell. It predicts age pretty precisely, at least as precisely as many other clocks. And it also shows a spread, which indicates there's still some variability between individuals. Interestingly, also, you can use this clock on another data set, which was not used to generate the data, on blood data, and it still works. So that was the good news. Now, obviously, the question that you're going to ask me, how does your clock and your new clock behave with respect to T-cell differentiation? And what I'm showing you here, again, on the left is the Horvath clock applied, again, to naive T-cells, central memory T-cells. And you can see each line corresponds to a different patient, a different donor, and you can see the increase in aging as the T-cell differentiate. And remarkably, our new clock is actually flat, so it doesn't change with T-cell differentiation. And in essence, really sort of fulfills the criteria of what we would call an intrinsic clock. And actually, we've called this clock entrant clock. Now, why is this important? So again, also measuring the effect, looking at T-cell differentiation, we used every other clock, Hannum, Horvath, Levine, and each of these clocks shows what's called a negative age acceleration, meaning there's a delta between the age of the patient and the measured age. And you can see in each of these clocks, actually focusing on effector memory T cells, you can see a delta, and our clock actually hits a 0, meaning it's not sensitive to this variable. Interestingly, it also works on CD4 T cells, so that was actually quite surprising, which indicates that some of these CPGs that were identified in CD8 T cells are likely to be shared in terms of the development of CD8 T cells. Now, where does this become important? You might have heard or read a number of papers that claimed the idea that COVID infection, for example, was reported to show accelerated aging. A 3-week infection associated with a 15 years acceleration of aging. And as a physician, I must say that I've always been extremely skeptical of the idea that you could age by 15 years over a 3-week period. And so, on the other hand, I'll remind you that COVID infection, or most viral infections, are associated with massive immune activation. So you have a lot of T-cell differentiation. And so the question is suggested that maybe what people are measuring under those circumstances is simply a massive immune expansion. Again, there's been the same type of data has been published for HIV infection. HIV infection is also associated with a shift from naive to more differentiated T cells. So we went back to this study, and you can see the previous clocks, actually Hannum, Horvath, and Levine all show deltas during infection with COVID. And remarkably, our clock actually shows absolutely no effect. It eliminates this variable, which really points to the fact that we would sort of confidently claim that many of these studies actually are measuring immune activation. Now, this also brings in an interesting other question is, can we develop another clock, extrin clock, that would actually measure this specifically? And Alan has developed one of these clocks. I won't have the time to go through it, but it's actually quite interesting. Now, one of the dirty secrets of the clocks, the epigenetic clocks, is that it's been extremely hard to understand what are the CPGs that are changing and how are they related actually to the biology of aging. And mostly there's been no correlation. When you look at these CPGs, it's very hard to discern what they are doing. What's been interesting is if you imagine that the current clocks are a mixture of 2 different clocks, and that's the data that I'm showing you, intrinsic, extrinsic, once you start separating them, the picture that emerges should be somewhat clearer. And indeed, when we look at the intrin clock, actually sites, we see sites that are more aligned with what many of us in the aging field have recognized as pathways that control the aging process. So I think that's one interesting aspect. I should say also that our clock, there's been a lot of talk about reprogramming and using the Yamanaka factors and aging. One of the interesting aspects of our clock is that it still really has a very strong signature in response to reprogramming. And on this data set, we've used methylation from fibroblasts that were present at different stages of OSKM reprogramming. And you can see that our clock actually goes back down to 0. Which is quite interesting. The other clocks don't go back to 0. They tend to go back to 15 to 20 years. Another piece of data I want to show you for 1 second is when we apply our clock, I showed you there's a very strong correlation between aging, chronological age, and our clock measurement, in all tissues except testis, which is remarkable. Testis actually shows, in our case, is an age that is stuck at around 15 years, which is right at the age of puberty. And I don't know if this really corresponds to the biology, but it's really interesting that that's the only organ that doesn't track with the other tissues. So in summary, what I hope to have convinced you is that CD8 T-cell differentiation state plays a major role in epigenetic clock age. We've created a new clock, which is resilient to changes in T-cell differentiation and shows some interesting properties. We're currently investigating the connection between this epigenetic age and measures of health span, transcription, and epigenetic changes. I would also finally repeat the fact that we think that interpretations of age acceleration or rejuvenation should be carefully reevaluated based on these data. And finally, probably the most interesting aspect to us is, what is our clock measuring? What are the genes? What are the pathways? Can we actually use the clock as a screening system to test drugs that actually are able to revert this? And so a lot of exciting new directions for the future. And I want to, again, thank here the members of my group, in particular, Alan Tomusiak, who is the graduate student who's conducted this work, but others, Herb Kassler, who's been helping us with the flow core, and others in the lab. Thank you.
Thank you so much. Thank you so much, Professor Verdin. One quick question.
Have you looked at chronic infections at all with the new clock, like HPV, HSV, Lyme disease?
Question is about chronic infection.
No, but obviously we are limited in some way by the availability of data sets. But there's a whole series of chronic fatigue syndrome, long COVID. These are all conditions associated with chronic immune activation, and I think, yeah, definitely interested in doing this or collaborating if someone has data sets that they want to share with us.
Thank you so much.
Thank
you
you
once again very much. Before I let